Hi, my name is Junli Leung. I'm in the microbiology class section 2965 of fall 2021. Today, I'll be presenting my Trichophyton rubrum pathogen. The pathogen names and classification. A scientific name of the pathogen is Trichophyton rubrum. The notable common names are athlete's foot fungus, jock itch, and ringworm. The pathogen is classified as belonging to the domain Eukarya, the genus Trichophyton, and the species T. rubrum. Here we have a Ramsey image of Trichophyton rubrum under a light microscope. The pathogen culture requirements. Trichophyton rubrum is the obligate aerosol, therefore it strictly requires oxygen in order to culture. Most of its fungi grow at a temperature ranging from 15 degrees Celsius to 35 degrees Celsius, with 32 degrees Celsius being the best temperature for optimal growth. In page 7 is best for optimal growth. Trichophyton rubrum grows more when incubating in the presence of light. Here we have an image of Trichophyton rubrum surface and immersed culture on Sauber round dextrose agar media after 30 days at 32 degrees Celsius at page 7. The pathogen actual size of Trichophyton rubrum. Here we have an image of Trichophyton rubrum on a fingernail from a transmission electron microscope, and the size of the organism is 8 micrometers to 50 micrometers. Pathogen life cycle. Trichophyton rubrum reproduced by the sexual life cycle. The cycle begins with a mycelium, which is a fungi in the vegetative form that consists of branch filaments called hyphae. Specialized hyphae, called coniophores, branch off from the mycelium and are able to produce spores. The next step of the cycle, the coniophores release their spores called conidia. The conidia spores are haploid and will undergo the process of mitosis. When the coniophores have complete mitosis, they will remain dormant to wait for ideal environmental conditions. Once the environmental conditions are favorable, meaning a warm, moist air and skin, coniophores will generate to produce a new mycelium and the life cycle will start over again. The sexual reproduction of a eukaryote such as Trichophyton rubrum transfer an identical copy of the genetic material to their offspring through vertical gene transfer. Therefore, sexual reproduction only introduces genetic variation into the population of a random mutation in the pathogen's genetic material that is passed on to the offspring. Novel structures produced by the organism. Trichophyton rubrum produce and secrete metallocarboxypeptidase proteolytic enzymes in acidic conditions as important valence factors. These protease enzymes have the ability to digest the keratins in tissues, such as nail and strand carnium, into sharp peptidases and amino acids that facilitate the invasion into keratinized tissues by Trichophyton rubrum. Trichophyton rubrum also produces and secrete endoproteases, which break down peptide bonds of non-terminal amino acids during host infections. Common names and diseases caused by the pathogen. The following are the common names and diseases caused by Trichophyton rubrum. Tinea pedis, also known as athlete's foot. Tinea crus, also known as jock itch. Tinea jugulum, which is nail fungus, also known as onychomycosis. And Tinea carpus, which is also known as ringworm. Where the pathogen causes disease and major reservoirs. Trichophyton rubrum frequently causes disease worldwide through contact with infected skin scales or contact with fungi in communal, damp, and contaminated environments, for example, such as swimming pools, public showers, and locker rooms. The major reservoirs of Trichophyton rubrum are humans and animals. Individuals most at risk. The individuals most at risk of infection from Trichophyton rubrum have the following: a nail injury, skin that gets warm and sweaty for a long period of time, or skin injuries in moist areas of the body where the skin folds. Individuals most at risk for serious disease from Trichophyton rubrum have the following: older age, blood circulation problems, obesity, and impaired immune system due to conditions such as diabetes or cancer. The mass transmission of Trichophyton rubrum are the following: direct hairless skin contact will contaminate warm, moist, and sweaty clothing, linen, and towels, or direct skin contact with affected skin scales that are shed, or direct contact with fungi or yeast spores when walking barefoot on damp floors in public areas, or direct skin-to-skin contact with an infected person or an animal. The purpose of entering Trichophyton rubrum is the cutaneous inoculation of fungi into injured, broken human keratinized tissues such as skin, nails, and hair that are often warm, damp, and sweaty. The incubation period is 4 to 10 days, depending on where the Trichophyton rubrum infection occurs on the body. The period of illness is the acute stage of infection, which lasts 1 to 10 days, and the contagious period of Trichophyton rubrum, the infection remains contagious for 48 hours. The signs and symptoms of Trichophyton rubrum infections are the following: dry skin, itchiness, flakiness, peeling and cracking the skin on the bottom of the foot, the groin, the thigh, the buttocks, arms, legs, and around the nails. Inflamed skin that might appear reddish, grayish, purplish. Red, scaly, elevated ring-shaped rashes in the brown arms and legs and buttocks. Clear, scaly area inside circular ring-shaped rashes with a scattering of bumps. Coloration nails range from colors from yellow, green, and black. And thick brown nails that chip or break easily. The tissues affected by Trichophyton rubrum are the following: the dust tissue called stratum corneum that's found in the outer layer of the epidermis is made mostly of keratin and lipids. The serious complications caused by Trichophyton rubrum are the following: fungal nail infections that cause the nail to be opaque, thick, whitish, and crumbly, and there may be inflammation and pain in the skin under the nail. Secondary bacterial infections that are painful, hot, and swollen. Cellulitis, which is a bacterial infection deep in the skin which can affect fat and soft tissues. And untreated cellulitis can lead to bone infection or septicemia, which is blood poisoning. The diagnostic methods for Trichophyton rubrum infections are the following: swabbing or scraping off a skin sample from the affected area to test for fungus by studying it under a microscope, or sending the skin sample or scrape of skin to a laboratory for fungal culture, or microscopic examination of affected nail clippings and debris from under the affected portion of the nails. The preventive measures for Trichophyton rubrum infections include the following: clipping nails short and keeping nails clean, avoid walking barefoot by wearing sandals or flip-flops in damp communal areas, keeping the skin in the groin area dry with a clean towel after showering or exercising, changing clean clothes after exercising, and showering immediately after each practice session or match in close contact sports. Here we have the narrative of a case study of a Trichophyton rubrum fungal infection called Tinea caput, which is ringworm in the scalp. A 63-year-old female sought medical care for a skin lesion on the central parietal region of the scalp that had hair loss and scattered scaly brownish papules after having the skin lesion for 5 months. Histopathological findings show follicular inflammation that affected the lobules, fat septum, and the septal hyphae of the hair follicles. After culturing the affected skin scalp scales on subarachnoid dextrose agar, the results revealed white-colored cotton-like colonies that had red-brown pigmentation on the reverse side of the agar. Trichophyton rubrum was confirmed as the causative agent as a result of negative urease results and the presence of multiple septate hyphae and microconidia. Under 30 days of systemic treatment, taking terbinafine 250 milligrams daily, and using ketoconazole shampoo, the patient recovered from the Trichophyton rubrum fungal infection and had new hair growth. Here's my sources slides, and that concludes my presentation of Trichophyton rubrum. Trichophyton rubrum overall is important in microbiology because a majority of dermatophyte fungal infections are caused by Trichophyton rubrum. That's why it's studied so extensively in microbiology. Thanks for watching the presentation. Thank you.